Hello and welcome to this updated for 2021 infection control lecture. My name is Peter Hampson and I am the Clinical Director at the Association of Optometrists. For this lecture, we're going to cover a range of topics. We're going to begin with a recap on the basics of infection control, just to make certain that we aren't slipping into any bad habits. We're then going to look at the routes of transmission of SARS-CoV-2, cleaning and disinfection, what works and what doesn't work quite so well, any updates from last time, and why these are important. We're also going to look at other steps to keep people safe, including social distancing, the effect of increased ventilation, the role that testing plays in keeping people safe, vaccines, and the effect that virus mutations may have on the effectivity of vaccines and the measures that we need to take. So if we turn to the basics of infection control first of all, we can see that there are really 5 areas: assessing the risk of infection, hand hygiene, PPE, good respiratory hygiene, and disposing of waste safely. And hopefully we're all very good at these things now, but I think it's worth just reinforcing what we should be doing just to make certain that those bad habits haven't crept in. So when we're talking about assessing the risk of infection, in a hospital setting, that would be determining if the patient has any particularly scary diseases. And in our case, that isn't quite the same. But it is about determining whether or not the patient that you're seeing is a high risk of having COVID or not. And it's about deflecting those that we think are high risk until they are safer, until the disease has passed, or until they have had a negative test and we know that they don't actually have it. And this is where the triage process is so important, because by triaging patients, asking the questions about whether they are well themselves, whether they have any symptoms of COVID, whether they've been exposed to anyone who may have COVID, or whether they've returned from an area where they should have been quarantining, they may have misunderstood the rules, they may have chosen to ignore the rules. But those processes will help to keep us and other people within the practice safe and should be the first line of defence for everybody. And they are therefore very important that those questions are always done before seeing patients. The next step is hand hygiene. And we should all be very familiar now with washing our hands thoroughly. I think most optometrists are already very good at it. But again, we've been doing it for so long, there is potential for people to become complacent, whether it is practice staff, whether it is visitors to the practice. So having the posters still displayed, making sure staff and patients are following those processes properly is really important to reduce risk. It's also worth bearing in mind that gloves don't remove the need for good hand hygiene. Just because you've got gloves on doesn't mean that you don't have to wash your hands. In fact, in the process of taking off the gloves, there is a risk of contaminating your hands, so it's important that we make certain that we maintain those processes. However, it is worth remembering that in the absence of gloves, good hand hygiene can mitigate the risk of not having gloves. So if you haven't got gloves to hand, as long as you don't have any open sores or broken skin, there is virtually no risk of contracting COVID through intact skin. And as such, if you are diligently washing your hands, then that lack of gloves can be mitigated. So PPE has largely remained unchanged. If you are working with patients at distances of 2 meters or less, then you should be wearing appropriate PPE. And there is a slight debate over whether we are medium risk in the new guidance or low risk in the new guidance, or the latest guidance I should say. But the medium risk sets out that we should be using single-use gloves, a single-use apron, type 2R fluid-resistant masks, and eye protection where there is a risk of exposure to bodily fluids. In the low-risk guidance, both aprons and eye protection are only suggested if there is a risk of exposure to bodily fluids. In our view, aprons still play an important role, even though they don't cover the shoulders and the upper arms, because it avoids the risk of transferring the virus to clothes, the legs, the sort of torso area, and all of that helps to reduce the risk of spreading the virus around the practice or spreading it to others. It's also worth remembering that PPE has a dual purpose. It isn't just there to protect you, it also helps to protect the patient by reducing the risk of spreading the virus around. Good respiratory hygiene. At the beginning of the pandemic, we all talked a lot about disposing of tissues appropriately, coughing into our elbow, sneezing into our elbow. It's still important that we keep those procedures up. There are many reasons that you can be sneezing or coughing that may be unrelated to COVID-19, and actually may be related to seasonal hay fever or similar allergies. But if you are unknowingly infected with the virus, if you are coughing or sneezing, there is a risk that you could be spreading that virus to others. And then the last point is making sure we dispose of any waste safely. And that can still be either via a Category B waste disposal contract or by double bagging the PP waste, storing it securely for 72 hours, and then disposing of it via the normal trade waste scheme. SARS-CoV-2 transmission broadly splits into two types: direct and indirect. Direct is that close-range transmission from person to person where someone is directly exposed to the respiratory droplets and aerosols, and that's what's classed as within the 2 metre zone. For indirect, we're talking where there is a contaminated surface and someone touches it and then infects themselves by touching their eyes, their nose, their mouth, or where there is the airborne aerosol that can be released by breathing, by talking, coughing, as well as by aerosol generating procedures, which thankfully are very rarely conducted in optometric practice, but are in their own right a risk. Since the onset of the pandemic, there has been lots of work into the factors that affect transmission. And some of this information is taken from the SAGE papers that inform the government decision making. And we can see that it's often not a single factor that is the key, it's a combination of factors based around the proximity, the duration of proximity, how many people are in the space, how well ventilated the space is, and then whether or not there are shared surfaces, as well as human factors, such as how often you are interacting with that person, whether you are mixing with lots of different people, which is why we've had this concept of small bubbles of people, small groups of people mixing is far safer, and also whether or not you can work from home or whether you have to go into a large office or a large workspace where people are coming in and out. In those larger office spaces, where perhaps ventilation is poor and you have a lot of people, the risk is far higher than somebody who's working from home. Also, for somebody who's working in a close interaction with somebody, such as we do in optometry, the risk is greater than for somebody who can always maintain a greater distance. Thankfully in optometry, the period of exposure to the person is relatively small, in that even though sight tests can be 25-30 minutes, that's a relatively short period of time to be exposed to somebody, especially when you consider that we have PPE. So according to SAGE, it is now thought that around 20% of SARS-CoV-2 infections are asymptomatic. And while those people are still infectious, it is thought they are around a third as infectious as those with symptoms. And if we think back to the basics of infection control, by avoiding the people that we know are symptomatic or are likely to be symptomatic, we are reducing our risk. Infectiousness is thought to peak at around 5 days. That doesn't mean people are safe after 5 days, but it does mean that there is a reduced risk. It has been noted that the PCR test, which we will talk about later, can be positive for a significant period after infection. And within the SAGE papers, the average was 17 days, but the maximum was 83 days post-infection that somebody could still test positive on a PCR test. When we think about indirect transmission, and we think about the surface-to-surface contact, what has moved on is our understanding of how long the SARS-CoV-2 virus can survive on various surfaces. So we now know that it's possible for SARS-CoV-2 to survive for up to 14 days on stainless steel surfaces, up to 7 days on nitrile gloves, and 21 days on an N95 mask. Interestingly, on cotton surfaces, it only appears to survive for 1 day. But obviously, most of the surfaces within a practice are a combination of metal door handles, hard surfaces. So there is a real risk there that the virus could be present for a significant period of time if we are not following appropriate disinfection procedures. Temperature also plays a significant part on how long the virus can survive for. And the sort of lower temperature has led to increased survival of the virus on some surfaces. And that explains why in the food packaging plants, where we've seen a number of outbreaks, where sort of lots of infections in a sort of a small group, it's a combination of the working environment, thinking back to those factors that affect transmission, as in how spread out people are, prolonged exposure, prolonged close working, but also the temperatures, meaning that the virus is surviving better for longer, and therefore there is a greater risk of indirect transmission in those cases. It was with this in mind that when we produced the initial infection control webinar, we looked at where we thought might be the common touch points within a practice. And we gave the obvious examples of clinical equipment, door handles, desks, keyboards, switches, keypads, anything where people may be in regular contact, including spectacle frames, where there was a risk of indirect transmission from person to person. There has been more research on this now, and a study taken from a Chinese hospital highlighted the regions or the areas where the virus was most often found. And we can see that the common touch points produced positive samples in a number of instances. So self-service printers, so where patients are printing off their own notes or badges or those sort of things, in up to 20% of those there was a positive sample found. Keyboards were another very common place to find positive samples, as were tabletops, door handles, telephones, things that multiple people are in contact with. And perhaps the things that if we aren't careful, it's very easy to forget to sanitize, because we may think, well, we've got a mask on or there's only staff using it, and forgetting that each staff member has various interactions with various patients throughout the day. And each of those interactions can produce a risk of passing on the virus, whether the patient is themselves infected or whether they have also transferred the virus from somebody they've been in contact with. It's also worth noting in the Chinese study that they also found positive samples on some medical equipment. So as much as there was good infection control, clearly at points things were missed. And it's important that we maintain those very clear, careful standards of making certain that the areas that we and that patients regularly come into contact with are disinfected. We've already spoken a little about the indirect aerosol-based approach, and this is one of the areas where our understanding has moved on quite a lot over the last few months. So aerosol-based transmission was originally taken as not very likely. We thought that SARS-CoV was just large droplets and that they fell to the ground relatively quickly. But a number of experts are now disputing that and disputing the basis for what classes as an aerosol or doesn't class as an aerosol. The original work on when particles were considered to be aerosols is based around some work by Wells from 1934. And Wells thought that when you coughed or you sneezed, you produced high numbers of respiratory droplets, and that is still fairly solid. But he suggested that based upon the size of those droplets, they either fell to the ground very quickly, or the fluids evaporated and then the residue drifted through the air. The cut-off point between when that happened was set at 5 micrometers. And it's this principle that is somewhat controversial. So a number of senior clinicians now dispute that that model holds true, and that 5 micrometer cutoff may oversimplify a very complex situation. There's been significant research that has shown that it isn't just the droplet size, but it is also the momentum or the force of the droplet that is travelling through the air and the surrounding airflow as well. So whether somebody is just speaking, whether somebody is whispering, whether somebody is shouting or singing, will have an impact on how quickly that droplet is ejected. And dependent upon how quickly it is ejected will depend on how long it may or may not stay in the air. And then the surrounding airflow will play a significant part in how long that particle can stay suspended for. So if you've got currents of air that may keep it floating at the upper reaches of the room, then that's going to have an impact on how long the particles are suspended for, and plays into the whole indoors versus outdoor risk. These considerations suggest that there's a far greater potential for respiratory particles to travel far greater distances than is initially thought, and why 2 meters alone may not be enough to keep us safe, and we may need to make certain that we're taking other steps. That also means that the protection offered by simple shields may be exaggerated, and that masks may be a far better form of defence. There has also been some concern with regard to aerosolization about how effective face masks are. But there is still very good evidence that suggests that surgical masks reduce the amount of virus emitted by a patient by 3 to 4 times, and they also reduce the size of that exhaled plume, and also the velocity of any particles that come out. Surgical masks further reduce incoming droplets and aerosols by an average of 6 times, and even homemade cloth masks reduce exposure by 2 to 4 times. So if both parties are wearing a mask of some form, the risk of infection is greatly reduced. There has been an argument that those treating patients with known COVID-19 might require more protective N95 masks where a fit test is required, so a respirator type mask. But within optometry, there is nothing to currently support this. In some instances where there are increased risks to the practitioner, it may be worth wearing an N95 mask as a belt and braces as a safe approach, but for normal practitioners there is no current indication that that is needed. Some of the other concerns around the virus have been that it's actually quite small, around 0.1 micrometers, and that may also mean that face masks do not work effectively. However, filters and face masks don't work on a simple sieve basis. Instead, they make use of a variety of processes to remove particles. These are impaction, interception, diffusion, and electrostatic attraction. These processes combine to trap particles as they pass between the fibres of a mask or of a filter, and small particles are captured very efficiently as they exhibit Brownian motion. So if we think back to our high school days and remember observing Brownian motion, as many of us will, and you can see that the particles move around in a fairly random motion, they don't travel in nice straight lines. It's that process that increases the rate of collision of particles with the fibres of the filter or of the mask. It's also worth considering that although the virus on its own might only be 0.1 micrometers, it is very rarely emitted as simply the virus. It's normally emitted attached to saliva or mucus droplets, and that significantly increases the size of the particles in which the virus is contained. So thinking of the virus on a pure size of the virus basis isn't actually an accurate reflection. So as much as masks and filters don't work on a pure sieve-like basis, it's worth considering the different mechanisms. So if they were working as a sieve in a straining mechanism, that occurs when the media or the particles are of a different size. So in the same way that if you pour vegetables or rice into a sieve, it doesn't fall through the holes because it's bigger than the holes. That's the basis that straining works on. But that isn't the main basis in which masks and filters work. But there will be a certain aspect of that is true. Impaction relies on the fact that the air is moving in a fast flowing stream, and as it hits the fibres it separates the particles from that airstream. So it relies on a direct collision. Interception doesn't involve a direct collision in the same way though. There is a physical contact and becoming attached to the fibres, but it works far better for smaller particles that aren't sort of large and just trapped by the filter media, and they sort of just glance against the filter media and are stuck to it because they're only very small. Diffusion, which as we mentioned just involves Brownian motion, because the particle doesn't often, or very small particles don't move in a straight line through the air, because they are sort of oscillating or bouncing around in the airflow, actually the odds are that they've got a much greater chance of hitting a barrier. And if you think of that as a car trying to drive between two bollards as an example, if the car is travelling in a perfectly straight line and it lines up with a gap between the bollards, it can pass through quite easily. But if the car sets about weaving from side to side and tries to just coincidentally get between the gap, the odds of that happening are far less. And that's what happens with the Brownian motion and the diffusion effect of filters. And then we have an electrostatic attraction. And in this case, ionizing wires induce a charge on the particles, which mean that the particles are then drawn onto the fibres of the collection plate. That's not quite necessarily always used in face masks, but it is used in some of the filtration devices that are out there, the air filters and the like, to filter the air as things pass through them. If we look at cleaning and disinfection, it's worth a brief recap again, just to remember the basics of what we're talking about. So cleaning physically removes infectious agents and organic matter such as soil. So when things are obviously dirty, you can't sterilise or disinfect an object that is dirty in the same way that you can't say there is visible dirt on a work surface or on a pair of shoes or anything like that, and that you've disinfected them. It just isn't possible. So it's removing the obvious soil matter from it. Disinfection is when we inactivate the pathogenic organisms, and that includes bacteria and viruses, High-level disinfection actually kills bacterial spores as well. And sterilisation is when we practically eliminate all viable microorganisms. And that doesn't mean we have to get rid of every single one, but it does mean that we have to practically get rid of every single one. And for a sterility assurance level, or SAL, we want to get a reduction of 10 to the 6, which means there's only a 1 in a million chance of an organism surviving. Again, if we think about how much we want to reduce the viral load by, we talked about log reductions and what is an appropriate reduction. And we used this graph before, and it still holds true that we want to reduce it by at least 99.9%. But I think it helps to visualise that if we think about the table as well. And within the table, we can see that for a 1 log reduction, we're getting rid of about 90% of the organism, whether that's bacteria or viruses in this case, up to a 6 log reduction, which is what we just mentioned with the SAL level of 99.9999%. And how that translates to the number of organisms is interesting. So if we had a fictional 1 million microbes, a 1 log reduction would mean that there could still be 100,000 of them on the surface, whereas a 6 log reduction would mean that there should only be 1. We're aiming for a 3 log reduction at least, which is 1,000. And that may sound like quite a lot, but as we're not particularly exposed to patients with active COVID, those viral loads aren't likely to be that high. And because of that, if we get rid of 99.9%, we've got a fairly good chance of surfaces being safe and secure and really reducing the risk of infection. When we think about disinfectants, we need to think about what still works or what doesn't work. So ethanol, sodium hypochlorite and detergents which contain sodium lauryl ether sulfate, alkyl polyglycosides and cocoa fatty acids are all still effective against SARS-CoV-2. Soaps disrupt the lipid layer and break the virus apart, and then they trap the parts of the virus and help them to be washed away. Alcohol works in a similar manner, disrupting the lipid layer, and bleaches, such as sodium hypochlorite, may also disrupt the lipid layer, but also attack the RNA of the virus, stopping it from working. Lots of people have asked about benzalkonium chloride, and a lot of us will be familiar with this as it used to be in contact lens solutions, and we may still see it in some drops, some artificial tears as a preservative. There have been a few studies that have been conducted on this, and in August 2020 there was a study that was conducted that suggested that benzalkonium chloride at 0.077 and 0.19% achieved a sufficient disinfection of SARS-CoV-2 in 5 minutes and 2 minutes respectively. However, a more recent study in November 2020 suggests that a 0.2% benzalkonium chloride could actually produce a sufficient disinfection in 30 seconds. As this is still sort of quite light on information and quite early days as to what does or doesn't work, we need to treat these results with a little bit of caution. So it would seem that 0.2% benzalkonium chloride is very likely to be effective at an exposure time of 2 minutes, and 0.1% should be effective at 5 minutes. They may work quicker, and that one study most definitely suggests that they could, but for caution's sake, we should base our disinfection times on those longer times just to make certain that a suitable disinfection is achieved. But the fact that the more recent study suggested quicker is suggestive that at 2 minutes we should be quite safe. So how does benzalkonium chloride work? We've touched on how soaps work and how alcohol works, and we spoke about those before. Well, benzalkonium chloride is a quaternary ammonium complex, and that also disrupts the lipid layer of the virus and helps to break it apart and to destroy it. One of the other things that we've had lots of questions about is hypochlorous acid. It's also known as hypochloric acid, chloranol, hydroxychlorine, and it's a weak acid that forms when chlorine dissolves in water and the chlorine then partially dissociates to form hypochlorite. Products containing hypochlorous acid can be found in the USA Environmental Protection Agency recommended list for disinfectants that are known to be effective against norovirus. The reason that the CDC places these sorts of cleaning products on their N list is that means that they may not have been specifically tested against SARS-CoV-2, but they have been shown to work against viruses that are known to be far harder to kill, than SARS-CoV-2, and norovirus is one of those examples. We know that norovirus can survive for incredibly long periods of time on surfaces, and that's why the winter vomiting bug, as it's referred to sometimes, can lead to outbreaks all over the place because it does just linger. Any disinfecting product that can disrupt that virus should be effective against the far less hardy SARS-CoV-2. We then need to consider how quickly hypochlorous acid may or may not work. And it appears that hypochlorous acid is effective at disinfecting surfaces following a contact time of around 1 minute, as long as we use a minimum concentration. And in this instance, it's 200 parts per million, which translates to about 0.02%. Weaker concentrations have also been shown to be effective, but the contact time is significantly longer, and in fact is 10 times as long when it's diluted by 10 times. So 20 parts per million would need a contact time of 10 minutes before it is effective. Hypochlorous acid can also be used in fogging machines to create an aerosol mist to disinfect an area. The fogging process can alter the physical and chemical properties of the disinfectant, and that may mean that the fogging machines need an adjustment to the concentration of hypochlorous acid that is used, and also that the pH of the solution must be adjusted as well. And this ensures that it is still effective. Most of the machines should have those adjustments included within their instructions so that people using them know what they need to set the machine to use. As long as that compensation is made, then fogging appears to provide an effective disinfection. However, as the fog requires a settling time to cover surfaces, to ensure that surfaces are also safe and it's not just helping to neutralise anything that is in the air, an extended time is required to make certain that the disinfection process is effective. And that may mean that fogging machines are highly unlikely to be suitable for in the day, but they may well be very appropriate for evening-based cleaning, out-of-hours cleaning, or for cleaning over a weekend, or that type of scenario. So how does hypochlorous acid work? Well, it works by binding with the lipid layer and disrupting the cellular integrity. Because it's effectively a bleach-based disinfectant, it also has that effect of damaging the DNA or the RNA of the virus. It is worth pointing out that all diluted disinfectants or cleaning products, once they're mixed up and made up into batches to be used, they should be used sort of straight away almost, but not stored, because once they're stored, the active ingredients can start to degrade and the effectivity of the disinfectant can start to wane. So if you're going to use these sort of products, it's worth making them up freshly and then using them for the cleaning process, but not being tempted to make them up in a batch in advance, diluted down, and then leaving them there where they may not be as effective when you come to use them, and the cleaning time may be far greater than you think it's going to be. So in summary, we now have this revised list of cleaning products. Ethanol has remained unchanged in concentration and exposure time, as has sodium hypochlorite, as has detergent. But we now have benzalkonium that at 0.1% we think will work in 5 minutes, and at 0.2% will work in 1 minute. And that hypochlorous acid at 0.02% should also be effective at a 1 minute exposure time. What's really important to remember is that if we're using detergents that need 5 minutes to work, then we've got to physically get that product onto whatever we are cleaning. So if we're cleaning between patients, we have to get the surfaces wiped down. We have to get that product on there, and that product needs a 5 minute exposure before you can wipe it off and dry the surface. So any ideas that you can be cleaning a surface or cleaning a room with a product that needs a 5 minute exposure time, and the whole cleaning process can only take you 5 minutes, cannot be accurate. You're going to need a longer time than that, because you've got to get it on, get the product back off and dried off again before the patient can use it, and then dispose of the cleaning wipes or the solution, or change your PP as appropriate, and all sorts. So it's very important that sufficient time is left between patients to actually allow that exposure time to happen, and that we're not just giving things a cursory wipe and not allowing for that correct exposure time. When we originally looked at UVC disinfection, we advised that spectacle frames are a complex shape that wasn't news to anyone, and that multiple light sources may help with UVC disinfection, but that people should be aware of their limitations and they should consider a belt and braces approach of pre-wiping the spectacle frames before using the UVC chambers, and also going for a long enough time to make certain that those safe disinfection standards were reached. We also advise that soap and water, as we've said, and detergents work very well. And we said that UVC is likely to be a useful tool when used with other disinfection techniques. Since we published that advice, NHS Scotland have conducted a rapid review of the literature around UVC. And their decision was that the evidence had limitations, and that meant that the mass rollout of UVC disinfection in a healthcare setting could not be supported at this point. They specifically commented that UVC air decontamination should not be used in place of optimum ventilation, and that making certain that there was an appropriate ventilation was actually important, not just trying to clean the air with UVC. But they did suggest that UVC disinfection might be helpful where there are aerosol generating procedures, again not particularly used in optometry but in dental practices and similar, to reduce the sort of fallow time, the gap between patients. They further said that UVC surface decontamination should not replace standard environmental cleaning and decontamination, that manufacturers should also make certain that they provide evidence of the effectiveness of their devices against SARS-CoV-2. So given the challenges around spectacle frames, we think that UVC disinfection can still play a part in disinfecting spectacle frames because they are generally quite delicate items, they are quite complex shapes, and that they might not be suitable for constantly dipping in bleach-based solutions because of the effect on colour, or it may have a degrading effect on the hinges and the joints if you're dipping them in and out of water all the while. So UVC may be appropriate, but to be safe, you should still go ahead with that pre-wiping of the frame with a suitable cleaning product to ensure that full disinfection is met, and then the UVC disinfection on top is effectively a supplemental technique to make certain that the true disinfection is reached. We've mentioned ventilation a few times now, and the main reason that ventilation is so important is this aerosolization risk. Aerosolized particles have been linked to this increased risk of disease spread because effectively you're working in this cloud of virus that's lingering in the air. And good ventilation is a key as it decreases that concentration by getting that air exchange and effectively diluting down any virus that is lingering in the air. That means that any exposure to the virus most likely means reduced viral load and therefore hopefully a less severe form of the disease or no disease at all. Natural ventilation such as opening windows works incredibly well, and as we move into spring and into the summer, that will be a very easy and very cheap way to make sure we get ventilation, having a window open, having the door open, and making certain that we get that really good airflow. Consulting rooms, though, don't normally have natural ventilation, and in those instances, local air cleaning and filtration units can be used to reduce the risk. The proper filters, the sort of HEPA type filters within filtration units, can help to reduce the viral load in the air by just passing the air through and effectively taking it out. And those filters work in the same way as the masks and the filters that we've already discussed. Some of those devices do include UV devices, and that's again another instance of where UVC disinfection can be useful, in that by bombarding the filter with UVC, it can help to kill any virus that is on there as well. So that can also then work really well in a mechanism that isn't necessarily as easy to do. That's quite different to the rapid review where they spoke about UVC air disinfection. This is actually as it passes through the filter. And a number of these devices are available, and as long as they have an approved filter within them, then they're not overly expensive and may represent a very sensible way to reduce the risk of viral load within the consulting room space. If you are looking at refitting, if you are looking at adding air conditioning, it's worth making certain that any process that you have has these appropriate filters. We should also add that the use of fans is fine when you have good ventilation already and good air circulation. But again, thinking about the aerosolization process, if you're in an enclosed environment, actually what the fan may do, if you haven't got good air exchange already, is it may just keep the virus suspended in the air for longer increasing that risk of an aerosolized environment rather than droplets where things are now on the work surface and you can wipe them down and clean them away. So if you can get air in, or if you can cascade air in one way or another through an open door and then force air out using a fan, that may be appropriate. But if the room is closed and there is very poor air exchange, then a fan itself may not be a good way of achieving that. One of the things that doesn't work very well is infrared temperature checks. And initially we thought that these might be a good way of checking to make certain whether or not the patient has an active infection or not. But what we now know is that because there are a significant number of patients who are asymptomatic, 20% or 1 in 5, it's not going to catch those patients. But also they're incredibly sensitive to environmental conditions. So on a hot day, the patient may be hot. They may have a hat on, so they may, their forehead may be warm because of having worn a hat or having run from the bus or whatever. And because of that, they can either give a false positive, or if a patient is asymptomatic or mildly symptomatic or pre-symptomatic, then the infrared temperature check may not catch the disease process, and they can lead to a false sense of security. And because of that, both Public Health England and the MHRA both advise against relying on temperature checks as a method of making certain that patients aren't infectious. It is far better to use the triaging questions and to look for the risk-based approach of whether or not somebody is likely to have disease rather than a reliance on temperature checks. We should all by now be quite familiar with lateral flow tests, as a number of us will have been conducting them in practice on a regular basis. And these are the ones that you could order through PCSE and have been provided by the NHS to practices. Lateral flow tests take a swab from either just the nose, as has been instructed by NHS England, or within the instructions of the test from the nose and the tonsils. And then that swab is dipped into an extraction solution and transferred onto the paper pad device that looks a little like a pregnancy test device. And then we get the results quite quickly. And it's about 30 minutes to get the results. They don't require any lab processing, but they do have limitations. Polymerase chain reaction tests, or PCR tests for short, are also referred to as molecular tests. And these actually detect genetic material or nucleic acid that is present inside the virus. They require lab analysis. The process involves viral RNAs converted to DNA, and then it's amplified by making millions of copies. The test then compares the DNA that has been found to that is known for the virus, and because of the amplification process, PCR tests can detect even very low levels of viral genetic material, but because they require lab processing, it takes far longer to get a result. PCR tests, however, are considered the gold standard of testing at the moment. And when talking about standards of testing, I think it's important to have a little recap on what we think we're measuring or what we are measuring when we talk about a test. So when we're talking about any form of test, whether it's a visual field test or whether in this case it's a COVID test, we need to consider the sensitivity and the specificity of the test. Now, the sensitivity tells us how good the test is at correctly finding the patients who have the disease. And in that instance, a false negative is when you test negative for the disease, but you do have it. A high sensitivity value means that there is a low chance of a false negative. Specificity tells us how good the test is at finding patients who don't have the disease. And in that case, a false positive test is when you test positive for the disease, but you haven't got it. And a high specificity value means a low chance of a false positive. Now, a perfect test would be 100% sensitive and 100% specific. But tests are never that good. So instead we have to look for high values for both to give us an idea of how good the test is. The World Health Organization suggests that for COVID, lateral flow tests should have a sensitivity of at least 80% and a specificity of at least 97%. Based on the initial data from the pilot in the Liverpool area, it appears that the sensitivity is actually only 48.9% and that the specificity is 99.9%. So from this we can see that if you test positive, there is a very good chance that you actually do have COVID-19. And that's where we are with the lateral flow tests. They have a very good success rate at finding those who actually have the disease when they test positive. So a positive test means that you most likely do have the disease. Where lateral flow tests are not so good is when it says that you don't have the disease, and that's where the 48.9% becomes relevant, because actually it may well have missed a number of people who do have the disease. And the Liverpool data looked at lateral flow tests and compared them against PCR tests, which, as we've said, is the gold standard. So what we now know is that if a lateral flow test is negative, you can't be certain that you do not have the disease. And therefore, while testing may help to find some staff who are asymptomatic and actually are carrying the disease, and it may help to break that chain of infection, a negative test does not mean that we should let our guards down or leave ourselves open to risk. We should be thinking that if we get a positive test on a lateral flow test, we should get a confirmatory PCR test to be certain, but actually we should be isolating because there's a very good chance that we do have the disease. But if we get a negative test, we still need to be very cautious. The other form of testing that is out there at the moment is antibody testing. And that looks at the blood samples taken from patients to see if they have antibodies that match and bind to the virus. These will be produced by people who have been previously infected, or potentially from people who have antibodies following vaccination. Obviously, as more of the population is vaccinated, the ability to look for antibodies purely from those who had the disease before becomes a little bit more difficult. So we're going to look a little at vaccines and what a vaccine is. A vaccine is a biological product that can be used to safely induce an immune response that provides protection against infection and/or disease. And vaccines have existed for a long while. Smallpox was 1798 when the first vaccine was introduced. Vaccines are used for measles, mumps, rubella. We've all probably had these when we were at school, or the vast majority of us will have done. And the types of vaccines that are used vary from weakened virus particles to where a whole virus has been inactivated or killed, through to particles that behave like the virus and produce a similar response. Viral vectors, where part of the virus that we want to protect against is inserted into a harmless virus that isn't going to cause a significant disease, so that we can deliver the mechanism via that and get the body to produce a response. And then more recently we've had mRNA vaccines, which is how the Pfizer vaccine has been developed. And those work in a slightly different way, but not vastly. They still stimulate this immune response and cannot affect DNA. There's been lots of talk about modifications to DNA based upon it, and that is just a physical impossibility of the vaccine to do that. The British Society for Immunology has produced some very handy guides to different types of vaccines that are out there. And I think it's worth taking a look at those different types, just to explain how they vary. So the first one is inactivated viruses. And in this case, you've got a killed virus, or in this case, it could be SARS-CoV-2, but it could be any virus within reason. And that killed virus is recognized by the immune system and triggers an immune response. And that response builds immunity so that our bodies can fight off the disease if we then encounter it. Sometimes they will need a booster, and that's the sort of virus that has been used previously for influenza. And there are clinical trials ongoing for COVID-19 vaccines based around that type of technology. We've then got attenuated viruses or weakened viruses. And again, there are trials going on for this for SARS-CoV, but it does take longer for these to be developed. So the weakened virus does the same thing. It triggers the immune response, doesn't give us the illness, so the virus is still active, but it isn't strong enough to cause a real or a significant infection. And then again, when we are infected with the actual virus, or if we are infected with the actual virus, our bodies know how to fight it off because they can already produce the antibodies so much quicker. And the polio vaccine is a good example of an attenuated virus that's used to produce a vaccine. We then have protein vaccines, and these contain the proteins from a virus. In this case, it would be SARS-CoV-2, and they're recognized by the immune system. Again, they trigger a response, and that's been used for things such as hepatitis B. They've got a very good safety record, and again, they may often need a booster. There are clinical trials ongoing for COVID-19 vaccines that work on this technology. Then we have viral vector vaccines, and these use an unrelated harmless virus, something like adenovirus. The genetic material of the virus that we want to vaccinate against, in this case SARS-CoV-2, is inserted into the other virus, and then that triggers an immune response. They may need to be stored at low temperatures in certain cases. And examples of that at the moment are the University of Oxford, the AstraZeneca COVID-19 vaccine. They use this sort of viral vector technology. And then we come to mRNA vaccines or genetic vaccines. And they contain a segment of the SARS-CoV-2, again in this instance, triggers an immune response. They are low cost, they are fast to develop, and that explains why we've seen this quick or apparently quick development of a vaccine compared to the attenuated vaccines that have taken far longer for testing. So the Pfizer vaccine, the biotech, and the Moderna vaccine are all examples of that mRNA. There is another one in trial at the moment, but that is what's led to this very rapid development of a vaccine. So what do we know about virus mutations? RNA viruses tend to evolve more quickly than DNA viruses. And this is likely how, or what helped at least, SARS-CoV-2 to move from bats to humans, whether it was via an intermediary or not. Each infected human will have hundreds of millions of virus particles within them. And within each of these infected humans, in this big pool of viruses, there will be a range of mutations. If one of those mutations makes the virus far more successful, then these mutations are likely to become the prevalent one. And that is what we've seen as we've gone through this pandemic, that we've seen some new variants become prevalent because they spread quicker or they're easier to transmit. And when we're talking about a mutation, what we're actually talking about is the spike protein. And that's the part of the virus that lets it sort of bind to the human cell, almost the key to the lock on the human cell. And a small or single change in that spike protein is unlikely to cause any significant problems. But as multiple changes occur, the chances that that vaccine may be effective start to decrease. So the greater the mutations, the less likely the vaccine is to be effective. And those changes to the virus may mean that it can just bind more efficiently. And that's what they think has happened with the South African variant and other variants. For now, vaccines seem to be working well, but it's already been suggested that it's likely that the vaccines will need an adjustment or a revision as these new variants emerge and as we move through this process. What's quite reassuring though at the moment is a number of studies have shown just how effective the vaccine is. And the Siren study, which looked at the effectiveness of the current mRNA vaccines against COVID-19 disease, and they found that actually the Pfizer vaccine against the B1.17 variant, when they looked at healthcare workers, they found that after 21 days it was around 70% effective, just from one dose, and that one dose was 70% effective at protecting from infection. After 2 doses that rose to 85%, but only once we got 7 days past that second dose. Public Health have also published a large amount of data on vaccine effectiveness. They've been monitoring the early impact and it suggests that the COVID vaccination is effective. GP data suggests that 60 to 70% vaccine effectiveness has been achieved in older adults just after a single dose. And obviously as people start to get those second doses, that will hopefully improve further. They also found that in those that had been vaccinated, even if they did go on to develop disease, they were far, far less likely to need hospitalisation, which is a huge positive. And in Scotland, there was a study that looked at data on the effectiveness of the first dose of COVID vaccines. They looked at the AstraZeneca and the Pfizer vaccine, and they found that both were effective at reducing COVID-19 related hospitalisations following a single dose. 28 days after the first dose was when the optimal effectiveness was reached, which is very reassuring that actually, even if people haven't had their second dose, after 4 weeks of being vaccinated with that first dose, there is a significant amount of protection in place. The AOP also has a number of resources available on the website that include face mask policies and posters that have recently been added to ensure that you don't inadvertently breach the Equalities Act and discrimination legislation if you're faced with a patient who cannot wear a face mask. Very worth checking these out. Thank you for listening.